There will be a Q&A for all panelists around 2:15 p.m. Eastern Time. Feel free to add questions at any time, but please bear in mind that we will likely not have enough time to answer all the questions live. And the moderator and panel will have that prerogative in discussions and topics discussed. I'd also like to highlight that the views and opinions expressed during this webinar are solely those of our expert contributors. Now, I'd like to introduce Jenna Jambeck, a co-chair of the Roundtable on Plastics and distinguished professor in environmental engineering at the University of Georgia. Jenna?
Thanks, Caroline. Welcome, everyone. On behalf of my Roundtable co-chair, LaShonda Corley, and I'd like to welcome you to this fifth and final webinar in our series on perspectives of the plastics life cycle. We have some amazing speakers today who are leaders in plastic pollution research and work, and I've had the pleasure of knowing some of them for many years. They'll share their knowledge and perspectives on plastic pollution and fate. And I'm thrilled now to welcome our webinar chair, moderator, and active Plastics Roundtable member, Imari Walker-Franklin. Imari is a research scientist at RTI International and also someone I know very well. So thank you so much for moderating today, Imari. I will pass it over to you.
Thank you all for having me here today. I'm honored to moderate this fifth webinar. So we know plastics represent a really complex set of materials that are formed from sometimes hundreds or thousands of different kinds of polymers. And it can be incorporated with also many kinds of additives and chemicals, forming many different kinds of products. This complexity can lead to further downstream challenges when it comes to the fate of plastic waste in our environment. So I'm excited for us to go further into these details with our first speaker on plastic sources, fate, and transformation in the environment, Kara Lavender Law. Kara is a research professor of oceanography at the Sea Education Association. Kara, thank you for joining us.
Thank you so much, Imari and Jenna, and to the Plastics Roundtable at the National Academies for inviting me to participate in this webinar. It's a great honor. I'm going to go ahead and share my screen and then get started here. Okay, so I am hoping to sort of set the scene for the conversation today by talking about plastic sources, fate, and transformation in the environment. And as I'm sure you all know, this is a really big topic, and that's in part because all the aspects of plastic pollution, their sources, distribution, and fate, and also impacts on wildlife and in humans really begins with environmental contamination. So plastics, this complex class of materials that Imari described, have been produced in increasing and immense quantities over more than 75 years, and they're contaminating the environment widely and broadly. So I want to start just with a little bit of sort of long-term perspective with a timeline highlighting the progression of our understanding of this plastic contamination in the environment since the nominal start of commercial plastics production in 1950. So the caveat is that I am an oceanographer, which my background will remind you, and throughout this talk, my focus is going to be on the marine environment. But through this timeline, I want to argue that there's a reason, another reason for this focus on the marine environment. And that's because I think really, to my knowledge at least, serious investigation into this contamination really began in the ocean. So in the early 1970s, we saw the first scientific reports of plastics floating in the open ocean, and that included both small bits of plastic and also larger identifiable items floating on the ocean surface, both in coastal regions, but also very far from land and human population centers. In 1975, the National Research Council of the National Academies published a chapter on marine litter in their Potential Ocean Pollutants report. And in this report, maritime inputs of all non-food waste, so all materials that were not edible, into the ocean were pretty crudely estimated based on the number of people living and working at sea. In 1984, NOAA held their first workshop on the fate and impacts of marine debris, again, the whole class of materials. The main focus at this point was really on fishing gear and other entangling gear, things like nets, cargo nets, also packing bands. And there was a big focus on the impact of entanglement and also some discussion on ingestion by marine life. In 1993, the Environmental Protection Agency got involved with a report on plastic pellets in the aquatic environment. So this was really recognizing that the raw material of plastics production was being lost and could be released into the environment through stormwater or combined sewer overflow discharges and spills during handling, including at ports and at sea. But it wasn't until really the early 2000s, I think in 2003, when this notion of a garbage patch in the North Pacific was introduced by Charles Moore in an article in Natural History magazine, that things really started accelerating in this field. That article was followed shortly by the first description of microplastics in a scientific article led by Richard Thompson. And shortly after that, we started seeing what I like to think of as the poster child of marine plastic pollution, which was photos of albatross chicks with bellies full of plastic by Chris Jordan. So since this time in the sort of early 2000s, I think we've seen a tremendous amount of attention, both by the public, but also by scientists, in documenting, at least at first, starting by documenting widespread contamination of all aspects of the marine environment, and then proceeding to the aquatic environment, the terrestrial environment, and the atmosphere. And this research continues fully today. So I can't ignore the fact that in parallel, there was a documented increase in contamination in biota. So I was describing contamination in the environment. Here, contamination in biota can include things like entanglement, ingestion, which is what this slide is demonstrating, and also other interactions with plastic debris. And so the first reports were actually in the 1960s of seabirds and marine mammals interacting with plastic debris. This plot starts in 1981, which coincides actually when I distinctly remember paying close attention to cutting apart plastic six-pack rings so that I wouldn't accidentally entangle a marine animal. So Santos et al. Put together this paper demonstrating the increase in peer-reviewed articles on plastics ingestion specifically. And in that article, they found that more than 1,500 species, including marine organisms from large charismatic marine mammals to fish to invertebrates, as well as freshwater organisms, terrestrial organisms, were having ingested microplastics. So that's just a little cue up for what's to come a little bit later today. So where we are today, we know that the Earth's 4 major environmental spheres are all contaminated by plastics. And I think it's fair to say that as new aspects of the environment were being investigated, the focus concurrently progressed from those large sort of megaplastics like those nets that I mentioned in the early reports to macroplastics, things like easily visible and identifiable plastics maybe floating at sea or on beaches, to the smaller microplastics and nanoplastics. And I think we can say, again, sort of safely say today that the smaller the particle, the higher the abundance in the environment. And that's at least down to microns in size. We still have some very challenging detection and identification challenges with nanoplastics in the environment because they are too small to be visually identified even under high power microscopy. So the planet is widely contaminated by plastics, but how much plastic is actually in the environment? So there are a number of ways that we can try to estimate this. First is by going into the environment itself and measuring it. And again, here, my focus is going to be on the marine environment. That is a bias. It's also what I know the best. So many of my comments will also be pertain to other aspects of the Earth's environment. One thing to point out before I go to the next slide is that how you try to measure plastic debris in the environment is determined by what size of debris you're targeting. Of course, you're going to need a different method to try to measure megaplastics than microplastics. So that said, I think to my knowledge, the largest number of measurements of plastic in the environment comes from studies using plankton nets. Plankton nets are very simple devices towed at the sea surface, or can be very simple. This is one that we use at my organization, Sea Education Association. These are designed to collect plankton, floating organisms at the sea surface, but they also collect anything else floating at the sea surface, whether it's large macroalgae or tar balls or bits of plastic. So in our case, we tow the net for 30 minutes, filters the top 25 centimeters or so of surface water in the ocean over a distance of 1 nautical mile, or just shy of about 2 kilometers. And in the late 1970s, students and scientists at my organization started finding little bits of plastic in the net. So the net comes aboard, you rinse the contents of the net into a sieve, and in a particularly contaminated sample, you might see something like this, where our students then visually identify particles that are pretty clearly plastic at these sizes. These are sizes larger than about 1/3 of a millimeter, and they count and archive those. So I'm arguing that most of the data collected in the environment on plastic pollution comes from plankton nets like these. And this slide is showing you the most recent publication on estimating the amount of floating ocean plastics collected by plankton nets. This is published by Erickson et al. A couple of years ago. And this map in the middle is showing you the locations of almost 12,000 plankton net tows over 40 years of collection. Well, this is just the latest of a series of estimates using plankton net data, and that's summarized on this slide here. So the first estimate was made back in 2014, and there have been subsequent estimates using increasing amounts of data in some cases, and also many different techniques. And so what you can see is that these estimates actually have quite a large range of the amount of floating microplastics. I put that in parentheses because, of course, that net is big enough to collect things that are larger than 5 millimeters in size, although that is more rare. So these estimates are ranging from as low as 7,000 tons globally up to almost 5 million metric tons globally. The variation is not simply because there's more data collected over time. There are different ways that people have handled the distribution of data, trying to account for different aspects of environmental variation or using different methods of extrapolating globally. Now, in contrast, I want to show a map of a different kind of data collection. So this is a map showing the distribution of microplastics collected in the aquatic environment, so in freshwater and seawater. And this is from a citizen scientist initiative led by adventure scientists. So in this case, samples were collected using 1 liter containers of water that were then sent to Abby Barrows, and who processed these using vacuum filtration through a 0.45 micron filter, and then visually identifying particles and counting them under a stereo microscope. So what she found was that 90% of samples contained microparticles, and 91% of those were actually microfibers, like you see in this image here. So the reason I'm putting this slide up here is to indicate that how we measure matters. The smallest particle that could theoretically be measured by this method of filtering 1 liter of water is about 750 times smaller than the smallest particles collected in a typical plankton net, like the ones I just showed you. So unlike the sort of large microplastics that are collected in the plankton nets, the naked eye can't easily discern whether particles at this size range are composed of plastic. And in fact, an analysis of a small subset of the particles in this study using micro-FTIR found that, in fact, 31% were not synthetic in composition. So we need to be very wary of how we are, or aware of how we are making these kinds of measurements. Okay, well, what about plastics in other parts of the environment? We know, first of all, not all plastic floats. So you might have some plastics that end up in the marine environment sinking to the seafloor. So this is from a study led by Alice Zhu to assess the mass of macroplastics, so now large debris, sitting on the seafloor globally. So what you'll notice, in contrast to the previous 2 studies I showed, is that there are many fewer studies. She identified 16 studies using remotely operated vehicles like the one shown here, and 25 studies using very large bottom trawl nets shown in this schematic. So what you can see from the map is that in this case, the samples are really highly concentrated in northern European seas and in the Mediterranean Sea. Even in the previous global studies, there were biases in where the samples were taken, but here you can see there's a pretty clear concentration north of 60 degrees latitude, and they were very sparse elsewhere. The other thing I want to point out is that the 2 different methods, using data from 2 different methods and then extrapolating globally, gives you 2 very different estimates of how much plastic macro debris is sitting on the seafloor. So the ROVs gave an estimate together with a statistical model, that's a generalized additive model, of 3 to 11 million metric tons, whereas from the trawl studies, there was a much larger range, but a much higher sort of maximum value estimated 571 million metric tons. Okay, what about microplastics on the seafloor? So I'm just pointing out, I should just say, these are not comprehensive reviews. I'm just pointing these out sort of as samples for discussion. So first, there was a study led by Barrett et al. In 2020 in the Great Australian Bight, where they collected sediment cores, 16 sediment cores, pretty closely together geographically, and then took subsamples from the sediment that was collected, and then extrapolated from the plastics they had measured in those subsamples to estimate that 14.4 million metric tons of microplastics are in seafloor sediments globally. So you'll see that sits sort of right in the range of the previous slide of macroplastics. A following study by Harris et al. Used data from more studies, and this is a little bit of a difficult map to see, but these little numbers here are showing you the locations of each of those studies that went into their analysis. So those are from 34 locations around the globe. And in this study, what they pointed out is that these measurements were actually collected in different sort of geomorphic areas of the global seafloor. So there were continental shelf, continental slope, hadal regions, hills, mountains, canyons, et cetera. So in this case, they had more data from more studies. So that means more measurements. But based on this distribution, most of the measurements were on the continental slope. And so instead of lumping them all together, they decided to take mean values of the measured microplastics in these sediments in 6 different categories of seafloor and then extrapolate within that category. And by doing that, which is taking into account sort of more information about the shape of the seafloor and how that might influence accumulation of microplastics, they found an estimate of 3 million metric tons of microplastics. Okay, so I'm just pointing these out because these studies have probably some of the most data in the marine environment of plastic contamination for both macroplastic and microplastic. Other environmental compartments, including large floating macroplastic or plastic of any size in rivers, lakes, reservoirs, the terrestrial environment and atmosphere, have far fewer environmental data. Okay, so that's just these studies are really using data at their core, and there might be some models or some statistical models used to make these global estimates, but there are other ways beyond just environmental data to try to make these estimates of plastic abundance in the environment. So first, a note about environmental data. As I think you probably can see, even in the best case scenario, these environmental data are sparse. So this is just the map based on sort of plankton nets. You see most of the world ocean is white, has been unsampled. Environmental data are also expensive to collect, might require an expensive research vessel to get offshore, or expensive equipment that is very time consuming to use in the laboratory to identify those smallest microplastics. And if you get closer to land and have to deal with a lot of sediment or organic matter, this is also a sort of time-intensive, labor-intensive process. But I argue they're sparse, they're expensive, but environmental data are necessary. They are not the only piece in the toolbox, the only tool in the toolbox, though. Numerical models provide a very important and contrasting view to studying this. So numerical models, as opposed to environmental data, which are sparse, numerical models are designed to give you a sort of complete solution. You get a number at every box globally, if you will. They're less expensive to run, and they can be as simple as one-dimensional box models, or they can have increasing complexity by adding additional processes to the models or by linking together multiple models. So I think numerical models are actually necessary, very necessary, to fill in gaps where we haven't measured the environment. They're useful to explore how processes interact, but the solution is only as good as the information available to build and test the models. So we do still need a fundamental understanding of both processes occurring in the environment to be able to parameterize those in the models, as well as environmental data to validate the solution. And then the third tool in the environmental science toolbox is remote sensing. So this, at this point, is a very nascent but exciting area of research, trying to measure from space or from aircraft on regular timescales, a global measurement. So at this point, I would say we're still in a very technically challenging area of method development to be able to detect and confirm the wide variety of plastic debris in different environments and against different backgrounds. And in the end, that will also only give us a surface measurement. Okay, of course, there are other approaches to estimating the amount of plastic in the environment. The next one I want to discuss is estimating it by estimating sources. So now we're thinking about not what's in the environment, but what's actually being input into the environment. And so just as an example, I'm showing you here a framework for sources of plastic debris, again, to the marine environment. This was initially conceived about 15 years ago by Jenna Jambek, who introduced this webinar. And then it was further developed by members of an NC's working group. You can find similar and actually much prettier schematics for other aspects of the environment, other parts of the marine environment or waterways, atmosphere, et cetera. In this case, we start with plastic resin, and we really conceived of the starting point as these manufactured pellets, which could be directly lost to the environment, but most of which would be turned into plastic products that could then be lost while they were being in use. So think about a hurricane or a flood washing items into the environment, lost fishing gear, lost shipping containers, et cetera, or they could be discarded and potentially lost. When we conceived of this, we hadn't really thought about direct pathways in use of microplastics to the environment. So I would now add on this little sidebar here where we have textiles, paint, tires, and probably other things that are actually being shedding microparticles into the environment while they're being used. Well, you can find many estimates in the scientific and gray literature for most of these sources, but I would say all of these are really indirect measurements. As difficult as it is to measure plastics in the environment globally, it's even more challenging to measure fluxes of plastics into the environment. And by that, I mean the amount that's being input per unit area, per unit time. So not every source has been crudely quantified, but I think it's likely that improperly managed waste is one of, if not the largest source of plastic debris to the environment. And I'm showing here a collection of estimates, a compilation of estimates of plastic waste input from land into the environment. This comes from a report from the National Academies on the U.S. contributions of global waste, of plastic waste to the global ocean. And what you're seeing here is a list of studies, starting with the study led by Jenna Jambek in 2015 and going through time. All of these followed the same basic framework that was presented in Jambek et al. And that's estimating first plastic waste generation, and then estimating the proportion of that plastic waste that was not properly captured and contained in a waste management system. So in this first study, we made calculations based on populations living within 50 kilometers of the coast. Subsequent studies extended that calculation inland along major rivers, and then eventually considering inputs into all aquatic environments, not only through rivers, but also smaller waterways, and acknowledging that debris contaminating ecosystems on land and freshwater ecosystems is also an issue. So at this point, or at least through 2021, these estimates of plastic waste entering aquatic ecosystems globally were as high as 23 million metric tons in a single year. But you can see that these estimates really do vary by orders of magnitude. We can take that a step back and say, all right, what about all ecosystems? Because again, you can get this sort of as a step in these studies, and these show that up to 99 million metric tons of plastic waste generated enters all ecosystems on the planet annually. I would not take any of these numbers as firm sort of final answers, but they do convey the scale of the problem. Okay, I'm running out of time, so I'm going to just quickly mention that most of the estimates of sources and reservoirs that I've been discussing represent either snapshots in time or long-term averages. But of course, we know there's immense variation in time in both the input and the distribution of plastic debris of all forms and all sizes. And this is just an example showing a 10-year trend in beach debris from surveys as part of the NOAA National Marine Debris Monitoring Program. This was particular plot is from beaches in the Northeast US and a paper by Rivik et al. And so you can just see that this is not a straight line. This is a nonlinear trend in both general source items, things that could be used on land or at sea, and items that were derived from the ocean. And a study by Smith and Markick actually demonstrated that the estimates of accumulation rates on beaches are strongly affected by the temporal scale of the sampling. So again, how you design the study matters. You'll get a different answer if you sample daily versus if you sample weekly. Okay, I'm going to skip over this in the interest of time, but want to make just two more slides, two more points. First, not only is there time variability in the environment, but we know that plastics are not static or entirely stable materials. They are altered by their exposure in the environment. So this is a schematic that sort of indicates a lot of these processes. First, we know that plastics do degrade chemically in response to UV radiation. They can also interact chemically with the environment by either leaching chemicals that are not chemically bound to the plastics themselves or by sorbing chemicals that are already present in the environment. We know that the photo degradation leads to enhanced sort of potential for fragmenting. That's what's generating many of these microplastics in the environment. And that's when experiencing sort of physical forces. We know that there is biological interaction with plastics in the environment. In the ocean specifically, there's biofilm that grows on the plastics. And of course, if they're eaten, they move throughout the food web. Mechanical breakdown can occur through those processes. And then below the dotted line, I like this graph because it sort of indicates, yeah, there is the potential for microbial degradation or assimilation or final mineralization, but we actually still know very little about this. And although it's likely that microbial degradation is very limited on any meaningful time scale. One thing that's not often discussed is this new evidence that plastics actually can photochemically dissolve. So by that, I mean, some of these chemical oxidation processes can cause the carbon in the plastic to be all the way broken down to carbon dioxide and other dissolved organic components. So this is sort of a new finding and doesn't necessarily suggest that plastics are going to magically disappear in the environment, but it doesn't mean that they're there forever. So I think where we're going in terms of degradation studies is we really want to sort of answer this question or find a metric to quantify how long plastics we're collecting in the environment have been there, how long have they taken to become microplastics, and how long are they likely to remain as plastics that are accessible to biota, for example, in the environment? What is their lifetime? Okay, finally, I just want to reiterate where what Imari started with, actually, that plastic is not singular. Plastics in the plural are a class of materials, and they're an extremely diverse class of materials in terms of both their feedstock, which mainly comes from fossil sources, but can also be bio-based. There are a wide variety of polymers. We often think of number 1 through 6 because those are stamped in our consumer products, but there are at least hundreds of polymers with highly specialized uses. They're further diversified by the chemical additives used for particular applications. And then, of course, innumerable product types, sizes, morphologies, et cetera. So everything that I've discussed depends on the specific formulation, the chemical formulation, the physical form, which then will determine how much is produced, how it's used, in what application, where it's used. That will inform its likelihood of being lost to the environment, how it's transported, how it's transformed, and how long it will persist. So plastics are a class of materials. That means they're also a class of environmental contaminants. And I'd say plastics were discovered and developed thanks to human ingenuity. We can't live without many plastics now, but we didn't really imagine the consequences of their persistence, their ability to weaken and break apart, and their potential toxicity. So that makes plastic pollution an extremely challenging environmental problem. With that, I'd just like to say thank you very much for your attention, and to all of my collaborators, both within SEA and especially through working groups that I've had the honor of participating in, which have greatly expanded my interest and understanding of the plastic pollution problem. And then thanks, of course, to all of those, especially federal agencies, that have provided funding to support this work. Thanks.
Thanks so much, Kara, for that wonderful overview presentation. I think we have maybe time for one quick question. But can you speak to what are some of the potential key technology gaps in this field that are hindering our understanding of fate or impact of plastic pollution?
That's a big question. So I guess I would say, my focus historically has been on trying to measure plastics in the environment. And I think we're not yet there for large scale sort of long-term monitoring. So for example, we really don't even have a great sense of how much large material, like this big net floating behind me in my image, how much of that is out there floating. And that's where the the remote sensing is a really promising technology. On the other end of the scale, nanoplastics are really challenging. We have a lot of evidence, sort of chemical evidence of the presence of nano or of plastic materials, but it's a little bit unclear, at least to me, whether it's sort of breakdown products that we're really measuring, or are we seeing polymers? Are we seeing particles that are still accessible to biota that they can, might be interacting with and experiencing ill effects from? So I think there's a lot of technological development at all scales that we would benefit from.
Oh, I agree. It's a big challenge for sure. Well, thank you so much again. So we will move on to our next speaker. I'm very excited to introduce Tracey Rochman. Tracey is an assistant professor in the ecology at the University of Toronto, and she'll be discussing the ecological impacts of plastic pollution. Tracey, thank you for joining us.
Thank you, Imari, and thank you so much for inviting me. Can you see my screen okay and hear me?
Yes.
Perfect. Okay, I will go ahead and begin. So I was invited here today to talk about the ecological impacts of plastic pollution. So I'm going to focus mostly on the impacts of microplastics and macroplastics, mostly to aquatic organisms, because I think that's where most of the data lies right now. So that's both freshwater and marine. But I will just say that there is more and more data coming out all the time that is relevant to the amount of plastics in terrestrial ecosystems and the impacts of those plastics. So I just want to note that that is relevant. I think Kara used the same statistic just to say that plastics are certainly, or organisms are certainly interacting with those increasing amounts of plastics that we're putting in the environment. And today there have been reported 1,500 species that have been reported to eat plastic. Now, this isn't because only 1,500 species eat plastic. It's because those are the organisms that have been studied, and plastic has been found within their GI tract. So it has been, at least in my experience over the course of my career, that when an organism is looked at in terms of does it interact with plastic, the answer tends to be yes, at least at the species level, that some animal, there's data on some animal that it has. And in my career, I would say I've seen an increasing amount of plastic in organisms over time. And I don't think this is because the amount of plastic necessarily in animals has increased over time, although that's probably true. For me, it's that I've changed the locations for which I've done my research, bringing myself closer to the source, and then likely seeing more ecological interactions and thus potentially impact. So when I started my career during my PhD in the middle of the first decade of the 2000s, I had an opportunity to go to the Great Pacific Garbage Patch, and Pete Davison and Rebecca Ash sampled myctophid fishes from the middle of the garbage patch, far from land, far from urban centers. And they found plastic in 1 in 10 fish, maybe a couple pieces of plastic in each fish where they found it. During my postdoc, in the early 2010s, I went and sampled fish from fish markets that were sampled offshore of California as well as in Vietnam. So again, these were coastal fish, open ocean, but coastal fish, where we were finding basically plastic in 1 in 4 fish, 25% of those sampled, and again, a few pieces in each fish. I then moved myself to Canada, where I work now on the Great Lakes, and it is a less dilute system in that they are, even though they are great, they are still lakes, and there's a lot of urban cities around them. And now when we sample fish from our Great Lakes, we find plastic in every fish, at least from Lake Ontario, and the numbers of plastic in the fish is much greater. So I want to show you some data from that study to just show that in an area where the source of plastic entering the environment is large and thus the concentration is large, the exposure can also be quite large. So I'm showing data from a study led by Maddie Milne, who was an undergraduate student at the time and is now doing her master's at the University of Manitoba. She received 6 species of fish from the Ministry of the Environment in Ontario. So these are brown bullhead, largemouth bass, northern pike, rock bass, smallmouth bass, and white sucker, for those of you that are interested in fishing. And here you can see that the concentrations or the amounts in each of the fish in the GI tract is on average about 100. You can see the medians here for some of those species that's quite high. And the highest we saw was actually more than 1,400 pieces in an individual white sucker. Now, a white sucker feeds from the bottom. And so based on its life history traits, it is more likely to eat microplastics, since a lot of the plastics over time do eventually sink. But the reason I really wanted to show this study is not just the idea that there's a lot of plastic in fish where the concentrations in the environment are high, but also that the exposure can also move into other parts of the body. So we understand now that microplastics can transfer into the muscle or the fillet of fish. And as we'll hear later in the third presentation, Tracey will talk about human health. And of course, this is exposure to humans from the fact that our mismanagement of waste has led to plastic pollution exposing our wildlife. Now, ingestion is not the only interaction that plastics have or organisms have with animals. We hear a lot or see in the news about entanglement. We know that organisms, for example, the sea turtle on the top right, can just have kind of this maze of plastic in their habitat that can lead to smothering, or in the case of this turtle, it can slow it down as it's moving from the nest to the ocean, which can make it more vulnerable to predation. I have a lot of interest right now in trying to understand how these plastics can actually act as a different type of habitat structure and potentially create new niches, or how organisms can interact with it, for example, for the bowerbird on the bottom right, as a form of mating display, which could affect sexual selection. So on the left-hand side, I have these images of how plastic can be interacted with by organisms as kind of habitat structure, or in the case of the hermit crab on the top left, a new home instead of a shell. So here I have images of organisms using plastics, for example, in the North Pacific garbage patch as a place to lay eggs where less structure might not be available if the plastic wasn't there. With fish in the Great Pacific garbage patch in the middle there, panel C, using it as habitat structure to hide perhaps from predators in the middle of the ocean, where again, that habitat structure is usually pretty low. And then on the bottom left, you see a picture of a fathead minnow in a lake using plastic as a nesting material. And those other two pictures are invertebrates using plastic as habitat. And for the foam in panel F, we saw a lot of macrobenthic invertebrates in that foam, which is interesting because that foam floats. And so it's almost moving those organisms to a new niche or a different part of the ocean. So a lot of questions about how plastics may interact with ecological processes and even evolutionary processes. I would say in the science, we're not quite there yet. So what I'll focus on today is what do we know about the impacts and how can we use that, or how have we been using that to inform risk assessment? Because we are there now, which is really remarkable. Several years ago, we just didn't have the amount of data to really make predictions about risk. So I'll start with showing data from systematic review from who used to be my PhD student, Kennedy Bucci, who's now Dr. Bucci and graduated. And she had updated a risk assessment that I had done during my PhD in an NC's working group, actually led by Kara. And here we were trying to pull out of the scientific literature the effects that have been demonstrated from plastic of all sizes across all levels of biological organization, from the molecular level affecting, say, gene expression, which is pretty easy to reproduce in a lab. It tells you something about mechanism. But if you have a change in gene expression, you may not have an effect that's ecologically relevant. To also those studies that have looked at how plastic impacts growth and reproduction, which may impact a population and in turn a community. So on the right-hand side, you're seeing those demonstrated effects that were in the literature as of, I believe it was 2019. So this is actually now kind of old. And on the left-hand side, or the y-axis, you see the levels of biological organization. And on the bottom, or the x-axis, you see the size distribution of the plastic. The darker shading means more evidence or higher weight of evidence. And so you can see here that by this time, we actually had quite a bit of studies demonstrating impacts of plastics across the size range and across levels of biological organization. I'll also say that we've started to understand more about the impacts or the mechanism of those impacts to organisms. While we still have, I would argue, a lot to learn, we do understand that those impacts can be both chemical in nature, say impacting the endocrine system and causing endocrine disruption, or physical in nature, causing, like you see here, entanglement, but also maybe causing an organism to feel full, leading to a food dilution effect where they have less energy, which may impact growth and reproduction. So next, I want to walk you through a study where we saw both. And again, I'm showing Dr. Kennedy Bucci's work here, where she had a full life cycle assessment running in our laboratory during her PhD, where she was looking at the impacts to fathead minnow, a freshwater fish, across their entire life cycle, from the larval stage through to adulthood, putting them in mating pairs, and then looking at their F1 or their offspring. She was using 2 different types of plastics taken off the shelf that were a cracked pellet. So in this case, polyethylene pellets that likely have some chemical additives in them, but maybe less chemicals associated with them than the polyethylene she collected off the beaches of Lake Ontario, grinding both down into a microplastic of the same size and shape, and exposing the organisms at 2 different concentrations, 200 particles per liter and 2,000 particles per liter. Now, these are environmentally relevant, but on the high end, and we only used 2 concentrations here. So we were really doing this hypothesis testing of what might be the effect mechanisms that we see, and do we see impact at those at these concentrations. We had 5 tanks per treatment, 5 treatments because we included a control. And here I'm going to show you all of the different endpoints we looked at and what we think we observed in terms of these physical effects and these chemical effects. So here you're looking at graphs that show you the effect sizes. You can see all of the impacts that we looked at are the endpoints on the left-hand column, and we've split them up into what we think is kind of this food dilution or this physical pathway and this endocrine pathway or this chemical pathway. You can see the different panels are for those different treatments. And the reason you don't see a control panel here is because these are effect sizes and the 0 in the middle of each graph is the control. And so what you're looking at is how the effect basically is compared to, like, how large the effect was compared to the control. And really only when it crosses that red dashed line is it considered biologically relevant. And where you see an asterisk, it is significant statistically via an ANOVA in this case. And so here you can see we did see biologically relevant effects in terms of growth and condition factor, which I think are related to that food dilution effect. But if you look on the bottom panel, you see effects that I think are related to endocrine disruption. Here we had broken eggs, likely due to thinning of the eggshells, which eggshells aren't in fish. I'm just using it to simplify it. They're the chorion of the fish, or that egg envelope. We also saw deformities in the F1 generation or the offspring, and we saw delays in reproduction. And we saw these effects more aligned with the lower concentration of the environmental plastic. And endocrine disruption, typically you see these non-monototic effects, where you see greater effects at the lower concentration but at the higher concentration. And I think the food dilution effect would likely have a more typical dose response curve. And here what I'm showing you on the top left is the number of eggs that were produced in each clutch and the day at which those clutches started to be produced. And you can see the treatments along the y-axis, and you can see here that the environmental low concentration started laying much later. And then when you look at the eggs of that environmental low concentration, you see more dead eggs or unfertilized eggs, but also more broken eggs, which we think is due to this thinning of the chorion, which you can see here in panel C is much thinner than what you see here in a control fish in panel D. And then finally, I can show you that in that low environmental concentration, we saw this higher level of deformities. And so here, I think this study does a nice job. I think it's showing that we have both these physical effects as well as these chemical effects from plastic. Now, there have been a lot of toxicity studies that have come out over the years, probably starting to come out in the literature around 2012, 2013, and today here we are at 2025. So you can imagine our weight of evidence has grown, and now we're able to put these studies into risk assessment frameworks. And so here I'm showing you a couple species sensitivity distributions, which are putting a lot of those different data from different toxicity studies together. So here you can see the percent of species that would be affected in this community of organisms that's included within the species sensitivity distribution, and the concentrations at which an effect was caused to each species. And here the effect can vary. Usually, like if you have, let's say, a rhizius here, this is a Medaka fish. If you have several studies creating that point, they'll usually pick the most sensitive endpoint and take the mean across those studies that caused a significant effect. So here you're seeing the species sensitivity distribution, which can be used to inform thresholds in a risk assessment framework. If a governing body was to say that the hazard concentration that affects 25% of organisms should determine a threshold, you could draw a line down and get at that concentration. So we've really moved into this space where, as Kara was talking about, it's really important to know exposure in the environment. And now we can actually compare those in a species sensitivity distribution in a risk assessment framework. And I want to give Bart Coleman's lab a lot of credit here in helping us figure out how we align that environmental data with the toxicity data, which I don't have time to describe today, but I could if there are questions. But now I want to focus a little bit on the macroplastic and point out some work that's been led by Dr. Erin Murphy, who's now a project scientist or a research scientist at Ocean Conservancy and was a postdoc at U of T with me a little over a year ago at this point. She's trying to figure out what are the best ways to combine data on macroplastic risk and be able to put these into some sort of model that can inform a risk assessment. In this case, we cannot dose macroplastic. We can't dose a lot of organisms with macroplastic. It's generally not ethical. So here we're using necropsy data. These are organisms that were found on beaches that were dead, and necropsies were done to see what was the cause of death. And so here we can look at how many of these organisms ingested plastic and how often was the death from plastic when plastic was ingested. So for data at a glance, these are pulled from the peer-reviewed scientific literature as well as databases of organisms that have washed up on beaches. Here you can see over 1,000 of each of 3 taxa: marine mammals, seabirds, and turtles, and there's 94 species within the mix and many different types of plastics found within the stomach. The other panels show you that within each type of taxa, the percent that consume plastic, the percent of those that consume plastic that had died from plastic in the necropsy, considering only that acute data where the cause of death would have been injury from the plastic, not necessarily emaciation, because we wanted to be really careful to feel sure that it was from that plastic. And here you can also see the top contributors that would have caused that harm in that organism. So for example, of the 1,430 birds that were found, 36% consumed plastic, and 5% of those who consumed it died from the plastic. And the top contributors of death in these organisms were balloons and hard fragments. Then Erin put all of that data into a model to try to be able to inform risk, here using a Bayesian logistic regression approach to predict the likelihood of mortality. And so here you can see for the seabirds, the turtles, and the marine mammals, using the data we could put in a model, you can see what is the percent likelihood of mortality from a certain number of species. Here you're looking at the means, and here you're looking at the 95% confidence intervals around that cumulative distribution function. So you could imagine this could also be used in a risk assessment framework where perhaps a 90% likelihood could lead you to a threshold for the amount of pieces on average that might kill, for example, one seabird. And so I think now that we've gotten here and we're able to create these risk assessment frameworks for micro and macroplastics, we really need to put our heads together and think about how can we use this information to inform monitoring and risk assessment in order to inform management. And also, I think it's really important, maybe not so much for the macroplastic, but at least for the microplastics, to try to ground truth those laboratory studies in the environment using mesocosm experiments or a whole lake experiment to try to understand, do those effects we see in a laboratory translate into nature? And what might be those ecological surprises from, say, indirect effects that we might see in food webs or even just within the community, within an ecosystem. So I'll spend my last 2.5 minutes just talking about kind of where we're going with this. And so in terms of the monitoring and risk assessment, working groups have been struck across North America. I'm sure this has also been done in Europe. A lot of the risk assessment work, as I said, a lot of the beginnings of that, the foundations were started by European scientists, but a California working group, as well as an International Joint Commissions working group between Canada and US for the Great Lakes, was struck basically in order to come up with what are the standard laboratory methods that can be used to quantify plastic, what are the standard sampling methods that can be used to sample plastics, and most relevant to today, what is a monitoring and risk assessment or a risk assessment and management framework that can be used to inform management. So I had the privilege to be able to co-chair both of these working groups. I've learned a ton, and I'll just show you a bit about where we landed with the IJC because this is our newest result. We had a report come out just about a month ago, and you can see here a link that will take you to that report. But just to give you an example of what you can do in different locations is you kind of take what is the normal management strategies that they use, say for contaminants in this region. And for the Great Lakes region, it's the State of the Great Lakes reporting and this 3-tiered management strategy, which would align well for 2 risk assessment or 2 thresholds for risk assessment. We can then create a species sensitivity distribution using relevant species and decisions made by stakeholders about what endpoints we're interested in, for example, as well as what do we think are those important hazard concentrations, what % of species do we want to protect, which is not a scientific question, that is a policy level question, coming up with those HC values and creating thresholds and putting those in risk assessment. So these risk assessment and management frameworks have been created, and then you can put them into these risk assessments initially with the concentrations that have been reported in the various Great Lakes, overlay them with the thresholds and see where do we stand now. So if you're just looking at Lake Ontario, for example, the dark green, you might actually put us into that fair category or potentially even into the poor category because several samples have been taken that actually do kick us over those thresholds. And finally, in my last 10 seconds, I just want to say we are in the process of doing a whole lake experiment at the ISD Experimental Lakes area in Northwestern Ontario. I don't have time to talk about it, but I wanted to say that this experiment is happening. We are using a lake. It is taking one for the team to try to help us understand what is the fate of microplastic, how does it impact ecosystems at all levels of biological organization, and then can we use that as a test bed to understand recovery? For example, Kara mentioned that degradation. Do we see this in our lake in a more controlled system? So this project is ongoing. Because the lake will take one for the team, we are answering as many questions as we can about every processes. There's about 22 PIs on this project and many, many students. So if you have questions about that, I'd be excited to tell you about it. But you can also visit our website here on the bottom left to learn more. And so with that, I'll thank all my collaborators and all the people I've learned from, and I'll stop there and see if I have time for questions now or later in the question period. Thank you.
Thanks, Chelsea. That's really exciting. Yeah, I'm gonna have to learn a lot more about that new project you all are working on. So I'd say for one question that we want to get at least before we move on to our next speaker is, do you have any idea why the lower concentrations of plastic contaminants seem to lead to higher rates of deformities or endocrine disruption?
Yeah, so in the endocrine disruption literature, it's actually quite common that you have these non-monotonic effects, meaning the effects don't tend to follow that typical dose-response relationship where the greater the dose, the higher the effect. Instead, you tend to see with these hormone-mimicking compounds, the lower levels tend to be the ones that cause effects. And this has been shown for DDT, atrazine, several other types of endocrine disrupting compounds. And this has to do with the way that the substrates interact with the enzymes. And so it has to do with the biological process at which the substrate will accept those enzymes versus start to push them away because they're overwhelmed. I don't know too much more than that. I would have to do that deeper research into that as well, but I will say it's pretty typical of endocrine disrupting effects.
Well, thank you so much, Chelsea. And then as a reminder to our audience, if you have any questions, please remember to put them in the Slido. So you go to slido.com and enter #plastic-pollution to join the conversation. So for our next speaker, I'd like to introduce Tracey Woodruff. Tracey is the Allison S. Carlson Endowed Professor in the Department of Obstetrics, Gynecology and Reproductive Sciences at the University of California, San Francisco, and the Director of the Program of Reproductive Health and the Environment. Tracey will speak on the human health impacts of plastic pollution. Welcome, Tracey.
All right, thank you. And I know it's a mouthful. So I really appreciate all the previous talks because I think it sets us up well to talk about the human health effects and what we know about the human health effects of plastics and microplastics. And I first want to say that I have no financial disclosures, and so therefore I have no financial disclosures to declare. So I want to pick up on what Kara presented in the beginning of her talk, which is that we've seen a tremendous growth in the production of plastic starting in the 1950s. And in particular, we've seen a growth in a doubling of plastic since 2008, and that plastic production is projected to grow or triple by 2060. So this is a really important problem that we need to address, particularly as you see, because it's contaminated many marine environments, terrestrial environments, and I will talk about the human health environment. So I want to also ground that these plastics are made from fossil fuels and chemicals, and this has implications for the life cycle of plastics, as well as who's exposed during that life cycle of plastics. And to pick up on also some of the discussion that Kara talked about in terms of what are plastics, what's in plastics. So there's the raw material that comes from the fossil fuel production, and there's monomers, but there's also a lot of chemical additives and processing agents. So there's a lot of different chemicals that are used, produced as part of plastic. So you have both the polymers and the plastic materials. And this report is from UNEP, and this is really excellent summary of the number of chemicals that are used in plastic production. The number that they cite is over 13,000. So those are chemicals that are in plastics, monitors, additives, or processing aids. This report also looked at some of the information, the toxicity information that there is available on these chemicals, these 13,000 chemicals that are used in plastics. And what they found was that there's about 7,000 of them that have been analyzed for their hazardous properties. And this can be something very simple as like maybe their structural properties, and that they've noted that there's 32,000 that are, or 3,200 that are of potential concern. And I think this is really important in terms of a number of different aspects. First of all, there's a lot of chemicals used in plastic. Second of all, we actually don't have good information about most of the chemicals that are used in plastic. But what we do know is there's a number of different types of chemicals that are used in plastic for which we have good toxicity information. So that includes flame retardants, perfluoroalkyl substances or PFAS, bisphenols, phthalates, polycyclic aromatic hydrocarbons, and then there are a number of metals, and then a number of other ones like UV stablers and biocides. And so this gives us some information in terms of understanding human health effects, which I'll talk a little bit more about. I also want to note that there's hazards all along the plastic life cycle. So first there's extraction. And so, for example, in the United States, we've seen a growth in the extraction of fossil fuels using fracking technologies, and that has led to the dramatic increase in plastic production since 2008. And so a lot of that extraction can also result in exposures in the community to those the various activities that are a part of the extraction process. And then there's plastic production. So during the plastic production cycle, chemicals are added. There's also emissions that come from these facilities, as well as exposures that can occur to the people who work in these facilities. Then there's the products that are made from these plastics. And so many of these chemicals that are used in these plastics can be released during the use of the plastics. And then there's the disposal. So disposal is often can be either dumping, I don't think talked about, but there can also be burning of plastic, which can release toxic waste. And so there's also potential for exposure to toxic chemicals during the disposal and destruction of plastic-related materials. And just to put more detail on that, you can see that there's a number of different chemicals that we at least know about during the plastic production cycle that we know are also toxic. So during production, you have vinyl chloride, styrenes, bisphenols. You also have air pollution coming from these facilities. And we know those are linked to a number of different different types of adverse health outcomes. Then during the use, as I talked about, there's many different uses of plastic. Plastics are so ubiquitous in our lives. They're used in personal care products, they're used in building materials, they're used in the various types of products that for example, cars, medical devices, and all of these different products contain these different plastic-related chemicals to which we are then exposed. And then in terms of the end of life, there's plastic-related chemicals that get into the dump supplies. You've heard already talks about the plastic-related, mostly microplastics that can contaminate the environment, like the marine environment and terrestrial environment. And then we also have that they can leach into multiple different venues, and then we can be exposed to them through the disposal, destruction, and dumping of these plastic-related materials. So I also want to emphasize that there are some communities have higher exposures to plastic-related materials and production due to their proximity of where they live near these different types of facilities. So this is an example in Louisiana where there are communities who live in these, this is from a report from ProPublica, but you can also see this from data that's been produced by EPA, that there's these locations where there's multiple different petrochemical industries, some of them producing many of the plastics that we are using in our daily lives, and that there is higher exposures in these communities due to their proximity to these plastic producing facilities. And I just want to note that there's been some estimates based on the emissions from these facilities as well as risk estimates that come from the U.S. Environmental Protection Agency, estimating the cancer risk of the people who live in these different communities, who tend to be on average poor, have a higher rate of poverty, and also are on average non-white populations. And those cancer risks can be up to 47 times higher than what EPA considers acceptable. So I think it's important when we're thinking about the life cycle of plastic is that the distribution of who's at risk is unequal. So the exposures to plastic-related chemicals, or I'll also talk about microplastics, come from all the ways that we interact with the environment. There's chemicals in our air, in the food we eat, in the water we drink. People can also be exposed dermally, and they can come from whether it's drinking water, food sources, but also personal care products, plastic materials that are used in the home, air pollution. And I want to also note that there, as I said, there's also opportunities for occupational exposures and people who can have higher exposures either due to their where they work or where they live. So we have been doing numerous studies related to biomonitoring of toxic chemicals in biological samples, and we have focused on pregnant women. We focus on pregnancy for a number of different reasons. The first is pregnancy is a really important time of great physiological changes, which also represent a time of higher susceptibility of exposure to harmful agents. So this is both true for we know that exposures that occur to the mother past the placenta and are also these chemical exposures are also measured in the fetus. And we've demonstrated that through numerous studies looking at matched maternal and cord blood samples, measuring chemicals that are matched maternal cord blood samples. So we know the chemicals are passing to the fetus. We also know that the fetal development is really important time for development because there's important and unique biological processes that are occurring during this time that only really occur during your lifetime and set the stage for the future child's health and even adult health. And disruptions to those processes during that time can result in adverse health effects, either in childhood or even in adulthood. And then lastly, the mother herself is also going through great physiological changes as she's preparing to both nurture the fetus as well as prepare for delivery. And those physiological changes, including increased blood production, development of the placenta, increasing respiratory function, and development of the mammary tissue, can also lead to increased risk from exposure to these different types of toxic chemicals. So one of the things that we've been doing is we were part of the National Institutes of Health Environmental Influences on Child Health Outcomes Consortium, and we had the opportunity to work with pregnancy cohorts across the United States. And as part of that, we expanded our capacity to understand exposures to these many different types of environmental chemicals, many of them which are also used or primarily used in plastics. And this is from a study that we've recently completed, and I'm just showing the results from the part that's focused on I would call the primary chemicals found in plastics. So we have here phthalates and phthalate alternatives. Phthalates are used as a plasticizer. They are used to make hard plastic soft and malleable. Parabens, flame retardants. These are often can be added a plastic additive. Bisphenols, which are used in plastic production, and then some benzophenones and antimicrobials. And what we did was we had a population of approximately 5,400 pregnant women from across the United States and Puerto Rico, and we measured these different chemicals in the urine from these women. And what this chart shows is that we found multiple different chemicals in their bodies. So there were 45 of these chemicals. This doesn't include all the chemicals we measured, because we also measured pesticides and polycyclic aromatic hydrocarbons. And we found 45 chemicals in over half of the pregnant women and 31 in greater than 70%. So the point of this is that we are exposed to multiple plastic-related chemicals, and that pregnant women are exposed to multiple plastic-related chemicals during this really important period of susceptibility. And this shows the percent of the population that is greater than the detection limit. So they had a detectable level of these chemicals in their bodies. And for the phthalates, you see that there are these multiple phthalates to which there are exposures, as well as these some of these bisphenols, and then as well as some of these parabens and benzophenones. And the other thing I want to say is that's unique about this particular study that we did was we measured some of the phthalates and bisphenols that people traditionally think of, like BPA or diethylhethyl phthalate. And then we also measured what's their kind of substitute, as there's been action on some of these chemicals like BPA because it's a known reproductive toxicant, or phthalates because it's known to adversely affect male reproductive development. We're also looking at what are the exposures to some of the chemicals that are being substituted for those chemicals in production as regulatory and industries respond to reducing some of these chemical uses because of their known toxicities. So you also get a range that we're also seeing exposures to some of the substitutes. So I'm going to talk a little bit about what do we know about the health effects from these chemicals. And before I do that, I want to briefly review how do we identify chemical toxicity. And I also want to talk about that there's a lot of people, there's a difference in terms of how the clinical community looks at these types of manufactured chemicals versus environmental community. And I think it's important to point this out because in the clinical community, when pharmaceuticals are developed for different types of interventions, there is actually pre-market testing that happens for those. So there's in vitro and in vivo toxicity testing, meaning they use cellular as well as animal testing to identify potential toxicity before they proceed to randomized controlled clinical trials to identify whether to ensure safety and efficacy of a pharmaceutical before it enters the marketplace. So there's a long history of using animal testing in clinical development of pharmaceuticals before they are prescribed to people. And environmental chemicals or plastic-related chemicals, this is not the case. That's because the laws that govern chemicals that were on the market prior to 1976, it says 62,000, 40,000 now, or new synthetic chemicals, primarily these chemicals that were on the market in 1976 when plastic production was well underway, they are allowed to be used on the marketplace, and there's essentially this kind of post-market testing on us that's occurring in terms of the way that the law works is that EPA is slowly prioritizing the chemicals, and then it's on the government to collect and identify the health information and then to act when they identify that type of adverse health effects in the population from these chemicals. So it's a very different standard. So environmental health evidence, there's been a lot of improvement in how environmental health evidence is evaluated. So we have both human and animal data, similar to the pharmaceuticals, but in this case, animal data is used because, as was mentioned by Chelsea, randomized controlled clinical trials are really considered unethical for environmental contaminants that can be toxic and don't have any efficacy properties. So the important point is that we have a long history of using animal data in environmental health. It's been this recent report by the National Academy of Sciences shows that did an evaluation of concordance of laboratory animals to human data, and that in general, laboratory animals have good concordance with human health hazards. Sometimes tests and animal data may be insensitive or not sensitive enough to identify certain types of endpoints. For example, developmental neurotox or mammary tumors. And that's because animals in these animal studies tend to be more homogeneous. They have a single exposure, they're not exposed to multiple chemical exposures. They're obviously not genetically diverse like humans are. They don't have differences in the population due to things like nutritional status or social stressors that people may suffer or have pre-existing conditions, human health, chronic health conditions, which we know is highly prevalent in the population. So the approach that is most rigorous and recommended by the National Academy of Sciences in multiple reports about how to evaluate environmental health evidence is a systematic review, which these are the steps here, but it's essentially a transparent, consistent approach to evaluate the quality, the strength and the quality of the environmental evidence to arrive at an overall conclusion about the strength of the animal evidence. And it evaluates both human data and animal data and comes up with an overall conclusion. And this has been, we developed one of the approaches in 2009, and now we are seeing the fruition of a lot of that because there's been a lot of use of systematic reviews have improved greatly our ability to understand the relationship between exposures to these plastic-related chemicals and adverse health effects. And this is a summary of what has been, what we know about some of these chemicals. I published this summary in the New England Journal of Medicine, but it really comes from these authoritative bodies, the National Academy of Sciences, EPA scientists who've done systematic reviews, and the California Environmental Protection Agency and the U.S. Environmental Protection Agency. And you can see that we have a number of different adverse health effects that are known are likely that are due to exposures from these different chemicals that are used in plastics, including perfluorinated chemicals, the phthalates, flame retardant chemicals, the bisphenols, and volatile organic compounds, and that includes things like formaldehyde or a benzene or vinyl chloride. And I want to note that these health effects are also health effects that we see increasing, many of them increasing in the population. So for example, phthalates are known to increase the risk of male reproductive toxicity. That includes effects on sperm. And we are seeing increases in problems with fecundity, which is ability to successfully achieve a pregnancy. The orthophthalates also increase the risk of preterm birth and metabolic disorders. Metabolic disorders are also increasing in the population with increasing diabetes and others. So I'm going to talk a little bit about microplastics. I see I'm almost at time. So microplastics are both from primary manufactured, but they're also secondary degradation, which the previous two talks talked a lot about. We know that these plastic fragments, as was previously discussed, are ubiquitous in the environment due to breakdown of the various plastics. And I think there was a discussion about microplastics, which are smaller than the human, smaller than the human eye. Whoops, I'm going the wrong way. Smaller than the human eye generally. So you have micro and nanoplastics, and that the plastic degradation of plastics is primary contribution of microplastics, which can come from packaging, textiles. I think this car tire wear is super interesting. We've been looking at this in terms of respiratory related effects, and then consumer products. So we're exposed to them through multiple, as I said, the microplastics, as you heard from the previous talks, they break down, they're measured in the aquatic environment, then we consume that aquatic environment. They also can get into our drinking water and then into the air. And this shows that microplastics, I just thought it was really interesting that there's been this increase in our understanding about microplastics in the ocean. And there's been, because of that we've had just an explosion in the amount of studies looking at microplastics, measuring microplastics in various parts of the body. There's been studies on microplastics in the brain, in the lung, in the kidney, in the colon. It's been measured in feces as well as breast milk. So we are exposed to microplastics in various parts of our body. So we conducted the first systematic review using these gold standard methods. We have for a select set of studies, health outcomes for microplastics. We reviewed 30,000 studies. We concluded that microplastics are a suspected digestive hazard with potential links to colon cancer. They're a suspected reproductive hazard linked to poor sperm quality, and that are suspected respiratory hazard with links to chronic inflammation and lung cancer. And I could talk more about that during the questions. But again, there's also how microplastics may exert these effects is still ongoing in terms of the investigation. We know that microplastics themselves are very similar to particulate matter air pollution, so they can, and we know a lot about particulate matter air pollution in terms of its known health effects. And microplastics could be an irritant, or it could be that these many chemicals that are found in plastics, which we know have toxicities, could also be contributing. So in conclusion, we know that chemicals that are found in plastics can increase the risk of these adverse health outcomes, and that's only a fraction of what we know. I just want to emphasize there's a lot we don't know about the chemicals used in plastic, but there's likely to be more health effects uncovered because there's so many more chemicals that we don't know about, and that we really need to address this growing plastic health crisis. So some of the areas where we're doing research is more rapid screening on health effects using in vitro technologies and some of these degraded microplastics, improving our biomonitoring and measurements in terms of biomonitoring, and focusing on how we can use existing policies to reduce the growing plastic production. I want to just end with these. First, to thank everyone that I work with at UCSF and outside. These are some of the reports that provide background on what I did in the talk, and you can follow us on all our channels. And thank you.
Thank you, Tracey. Hold on one second. We are about to slide back into a questionnaire, but give me 1 second real quick. So thank you again for your presentation. Would you mind speaking a little bit from your perspective about what is the best approach for addressing the microplastics problem?
Which part of the problem? Are you talking to me or all of us?
For you, Tracey. And
oh, yes, what's the best approach to talk about microplastics problem? The approach is approach in terms of our personal approach, our policy approach. Did you have a thought about that, Imari? I will just say, I talked to a lot of press about this because people obviously, they're super interested in this microplastics issue, which is very understandable. And I would say that we've been both talking to people about, well, there are things that you personally can do to reduce either it's chemical exposures, which these plastic-related chemical exposures or microplastic exposures, which we offer our recommendations on our website. But we really encourage these. I think what the other speakers have also focused on is these more structural solutions, because the concern is that we have a lot of plastic and there's this projected increase in plastic. So whatever problems we have now are only going to increase. So that's why policy solutions are really important in this case.
Absolutely agree.
And now I'll say that we are about to enter our moderated portion of our conversation. So thank you again to all the speakers. If you all have any questions, please remember to navigate to slido.com, enter hashtag plastic pollution to join the conversation. So I'd like to now invite all of the speakers again to join us for Q&A. And you can also upvote any questions that you're most interested in the Slido as well. So for the first set of questions we'd like to go through, well, we'll throw it back to I say this could be for anyone that would like to answer, but mainly maybe Tracey first. With even more known data on the effects of plastic contamination on organisms, where are you now on recommending or classifying plastic waste as hazardous? And would this still be a recommendation from like maybe working groups to manage plastics?
Plastic waste is hazardous?
Or
Well,
even microplastics?
yeah, no, I mean, we definitely think that there's a hazard to microplastics. The exact nature of that hazard or there is uncertainty about it, I will say that. So I think that it's important to understand, there's the part about the science, which we all talked about the science, and then there's the management of how do we address that in a context for making decisions about these about microplastic or plastic related waste. And I think that it's really important to a caveat that, when you have enough information, because what's really I think what Kara's presentation showed really excellently is that we're just going to get more information about where these are accumulating. We're going to get more information about their exposures, and we know we're exposed right now. So it's really important to take action because I think there's someone from the head OMB used to say, I'd rather be vaguely right than precisely wrong. So if we wait too long to address with this, and I want to say something about this too, that just to follow up on the persistence point, we already know from our experience with DDT and PCBs, we still measure DDT in our populations here in California, and that was banned 50 years ago. So it's for these pollutants or these microplastics, which are persistent and bioaccumulative. I mean, maybe they may degrade sometime in the way far future, but we know we're going to have a problem for a long time. And that's why it's really important to take action on those types of chemicals and hazardous constituents sooner, because if we're wrong about the toxicity, it's going to be, if we're wrong and it's really way more toxic, then we're going to have a really even bigger problem.
Absolutely. And then Chelsea, would you like to speak to classification of plastic waste or microplastics as hazardous?
Sure. Your question made me giggle a bit because when I was in my PhD with several colleagues, including Richard Johnson, Hideshige Takata, others in the field, we wrote a paper, an opinion in Nature about labeling plastic pollution as a hazardous substance. And it got turned around into plastic waste as hazardous, which is never what we meant. So I think we do have to be careful about what it is we're labeling as hazardous. But I do think there's enough data today to suggest, I agree with Tracey, that microplastics and plastic pollution are hazardous in the sense that they are persistent. I think we know they can translocate into organisms. I think for the size of plastics I focus on, we're still trying to figure out how bioaccumulative they are, but we absolutely also know they can be toxic. So I know that in the Great Lakes region right now, they're looking at a proposal to label microplastics as a chemical of mutual concern, for example, which would turn on a lot of those same regulations that hazardous substance labeling would do for microplastics as a pollutant under the Environmental Protection Act. So yeah, I agree with Tracey, and I also just want to talk about the history of that question because that has been going on for a while, and I think we have to be quite careful of the words that we use. And I understand that even the paper I wrote had started that by using the wrong words, so by being misconstrued by the media.
No, I think that's great. The distinction between waste and pollution itself. So then I guess to follow up into another question, We talked, a little bit about DDT you mentioned, Tracy, and just the timeline that we talk about getting from science to the final result of action. Maybe we could start maybe with Kara too, but how do we shorten this timeline between scientific investigations, risk assessments, management regulation to action on the ground? Do you think there's any tools that we could use or, to get us going? And yeah, whoever might want to have the answer first.
I don't know if Kara wants to go first. I'm happy to also add.
I can start with some thoughts, I guess. So in the National Academies report on the U.S. role in global ocean plastic waste, we presented an intervention framework of sort of how to address plastic pollution broadly. And it's not a magical framework. It talks about reducing plastic production at one end and environmental cleanup at the other, with a number of steps in between. And I think there are already many actions happening along that entire spectrum, and they all need to continue happening. This problem's not going to be solved entirely by regulation or entirely by policy. I think there is a lot that the sort of individual can do to either reduce their own exposure or to prevent the sort of leakage to the environment. I heard a great webinar put on by Bush Systems, which actually makes recycling bins in Canada. And one of the people was talking about a park in San Francisco, Dolores something. But it was a whole campaign about sort of observing why the park was becoming so littered and then figuring out how to address those levers, right? So some of it was public outreach, some of it was working with local businesses, some of it was moving where the trash cans were or the recycling bins, and some of it was signage. And I just loved it because it was really a great case study of a very specific instance of like identifying the problem and the sort of drivers of that problem, and now there's this beautiful clean park, right? So there's probably parallels to that in all aspects of this pollution problem. I'm not going to speak to the health and impact side. That's not what I do. So I'll pass the baton to the other two now.
Yeah, so I think that's a really important point that you raised, and I think there's different ways to try and shorten the time between the science and the action. And some of that is, I think we the way we think about it is there's working within the legal frameworks that we have, which and then recognizing that some of those legal frameworks have flaws in them and that they need to be, they don't truly address what we need to do in terms of preventing these types of, particularly these persistent and bioaccumulative pollutants getting out, or some of their better legal frameworks, for example, in some of the states. So I think in terms of just identifying the hazards, that's one reason why we did this work to improve the ability to evaluate the hazardous properties of chemicals, whether it's chemicals or microplastics, through this systematic review process, because technically it provides a more consistent approach to how the evidence is evaluated with less bias. And that provides a bottom line summary that policymakers can use to take action. I mean, our bottom line summary is suspected, but let's just say, would you give your child something that 50% might cause them to have some adverse health effect? Most policymakers would say, no, I don't think so. So I think that's a really important component. But I think the more upstream part of this is that we really should be focusing on these chemical properties like persistence and bioaccumulation, and that those should have a particular focus in terms of banning or removing from basically production. And we know from data on DDT, on perfluorinated chemicals, on air pollution and lead, that when you take those substances out of the environment or you reduce the production or emissions of them, the levels in the environment or in people go down. I mean, the classic example is removing lead from gasoline meant that people in the United States had much less, children in the United States had much less lead levels in their body, to levels that people thought actually were not even possible back when lead was in gasoline. And even though we still have DDT, it's definitely, the levels have gone down. PFAS, same thing. So I think we just need to, we're scientists and we look at the science, but we really have to engage with other actors in the in our working in this space to engage with those who are promoting policy solutions that will really be systemic solutions to this problem. Because I agree, there's definitely great evidence about you can reduce your personal exposures to many of these chemicals, but you can't get rid of it. And that's partly because there's some of these exposures you can't control.
Oh, absolutely. And I guess, it's going to take a lot of work to kind of get us to that final level of policy analysis for sure. But,
Right.
the further we get there, the better. So one of our next questions from the Slido had to do more with, I'd say Chelsea's talk on, I think you gave us a teaser of the work that you're doing with the new lake study with microplastics. But I think they're wondering also, are there additional tools or websites that the public or scientists could use to further our understanding of the impacts or the fate of plastic pollution?
Yeah, so I'm going to answer the second part of your question, which is I think you're asking, are there tools that are available that the public can go to understand impacts? And so what I didn't have time to say and had originally had a slide and had to throw it out is that as part of that SCORP working group in California, so the database I showed of the Bucci paper from 2020, where it shows all of those effects, those were all updated by Leah Thornton Hampton and colleagues in 2023, I believe, with the paper. And then actually that database went into creating a publicly available open access Shiny app called Tomex. And anyone at any time can go into this Tomex database. It's about to be updated to 2.0. Right now, it's still 1.0. But essentially, you can go in there and look at the patterns of all the toxicity data and also build your own species sensitivity distribution. So the idea is that any government, anytime, could go in there and think about what might help me build my relevant SSD, and then how can I build that species sensitivity distribution and use it. Now, there's a lot of, I think, working groups being struck to help make those decisions is a good idea. The government's going to go then take it and use it to inform management. But that database should be ideally continually updated. We're trying to make sure we have resources and support for that. But so far, that has been really great. We did create a version for human health. At the time, the human health data was pretty small. Now there's a lot of researchers working in that field, which is amazing. And so eventually that database will also be able to function well for human health. But at the moment, to the best of my knowledge, there's no kind of risk assessment framework that's been created yet for human health, although Tracey can correct me if I'm wrong. But we tried to do that with the SCORP working group, and in 2021 decided we didn't feel we had enough information yet.
And then, Kara, was there any other websites or tools that you know of that help with plastic pollution, maybe tracking, or awareness of debris in the ocean? Muted.
I know, well, yeah. So there's UNEP's GPML database. I mean, I think there are places that are trying to collate data in the environment. It's increasingly difficult. Certainly when Chelsea and I first met, and this field was sort of coming out of nowhere, not out of nowhere, but it was kind of, there were not a lot of people working on the field, and it was mostly ocean. You could sort of keep track of what was happening. Now the literature has just exploded in all these different environments. And so I think there's probably many efforts that I'm not even aware of for people who are interested in microplastics in the atmosphere, for example, or in terrestrial systems. So I don't have great advice about where to look for those kinds of compilations. I think it's a very difficult thing to stay on top of.
Can I add one more thing? Because I wasn't thinking of it until you asked Kara to follow up. The TIDES database through Ocean Conservancy is an amazing resource for open access global data on cleanup data. And cleanup data, of course, is community science data that is not always as quantitative as you might like it, but it's actually pretty good and has been used in dozens of scientific publications. And so that TIDES database is almost a 40-year database of cleanup data that now includes data from the International Trash Trap Network, so both the trash traps around the world and the cleanup around the world. And that could certainly be used to help inform monitoring for at least macroplastic.
That's great. Thank you. And I guess another one from the top of my head is the waste debris tracker that Jenna Jembeck's group started. And then I think Literati, they also have apps for public if you want to engage with, doing a beach cleanup or a waste cleanup and categorizing that data or even getting access to it. So that's really great. Moving to another idea of just, and an EPA mapper as well. Moving to just like idea of sources or resources that we might be limited to. So for Tracey, could you speak to, how we can monitor plastic pollution health effects or microplastic health effects in a resource limited setting? And I think they're speaking more to maybe grant resource limited.
Yeah, okay, resource limited setting. It's like, so I think is the question, how do we do more studies on microplastics? Anyway, I mean, so currently, obviously, there's a lot of, one of the challenges that we face as researchers is there was just some memos leaked, were reported in the New York Times and the Washington Post about how there's proposed pretty big budget cuts coming from the White House to National Institute for Environmental Health Sciences, which is the primary funder of research in this area. So, that doesn't mean, it's obviously Congress. Well, typically Congress would make the decisions about funding in this area. And so I think given that there's so much, the thing that's really important about microplastics is that it's really it's almost like an aggregate. I like to think of microplastics like I think about particulate matter air pollution. And so people who've done work in particulate matter air pollution, I don't know, to people on this call, maybe we know a lot about particulate matter air pollution, and we know, and that is because there's requirements for monitoring for particulate matter air pollution, which people have linked to many different types of health databases. And we know so much about particulate matter air pollution, including that it increases the risk of death, cardiovascular disease, neurological diseases, adverse pregnancy outcomes. It's just a wealth of what we know. And particulate matter air pollution is particles that are less than 2.5 microns. Microplastics are less than 5. So, microplastics are part of that whole gamish of things that are together. And so I think, to me, I think the study of microplastics is really important because it really aggregates a lot of different cumulative or aggregate exposures to many different aspects of plastics, both the chemicals that are in the plastics as well as the structural microplastics themselves. That being said, the research landscape is extremely, I'm going to use a kind word, is uncertain, but probably scary. People who are at universities right now are very worried both about the issues related to even what's the overhead going to be, which provides that really essential money to keep all the buildings running so we can do the research, to propose cuts to the research that we do. So that's definitely resource limiting. I don't really have a good answer. Some of the states, I can just speak, I'm in the state of California. I know there's been legislation introduced in the state of California to create science funds to help support research in the state of California. It's hard to imagine that can be at the same level. The federal government really plays such an important role in providing the data that we need, not just on microplastics or all these types of things that we work on, but just everything related to addressing health. So it's kind of a hard question to answer.
Oh, I understand.
Maybe
I
the
think
people
we're all
in
figuring it
Canada
out.
have an answer. Maybe we are going to move to Canada. Maybe that's what we'll do.
Well, I guess maybe a little bit more focused then. From your systematic review, I think a lot of the papers that ended up narrowed down focused a little bit
Yeah.
more on one type of plastic, right? It
Yes.
was mostly polystyrene. So like, what would be the next step? If somebody was to
Yeah.
redesign a human health type of study, what would you recommend?
Yeah, so I just want to say that What was interesting about those studies, first of all, none of them were done in the United States. That was very interesting. So a lot of the work, I think it's a little bit like the ocean work. We're seeing a lot of this work happening kind of in other places and then slowly migrating to the United States. And then we really focused on studies that more mimicked the human experience. So they were animal studies. That was almost all the studies have been done to date. And that they had to be exposed chronically, similar to humans, so that they had not just one exposure, but multiple exposures. And then they also we only looked at certain types of health endpoints because it was limited in terms of the time we had to do it. And then we focused on those health endpoints we thought were going to be most relevant to the routes of exposure. So that is looking at the digestive system, the respiratory system, and then a sensitive endpoint, the reproductive system. And it turns out that all the studies to date have basically purchased microplastic beads. That's what they went to Sigma and bought the microplastic beads and then exposed the animals through not gavage, but they had to be drinking it or in their food. So we also only looked at those studies that mimic the more like a human experience. So to that being said, we don't really know, and they were microbeads, they weren't nanoplastics, they weren't degraded plastics. So there's a lot to do in terms of adding more types of plastic to either animal studies or looking at how to measure microplastics in humans, which you can measure the microplastics using some of the technologies that Kara talked about, but there's still a lot going on in terms of how do we measure these smaller nanoplastics with, some of the details and the methods. And so that's another area that we've also been looking at, and maybe people on this call have some ideas. We've been looking at some very interesting areas. So the measurement is also really important. And I just want to point one more thing about the microplastics, which is we found very interesting, is it's not just, because some people just take plastics and grind them up in a grinder. But as Kara talked about, that's not how microplastics come about in the environment. There's like sun and they're washed around in the ocean or whatever. And so trying to figure out how to weather microplastics that people are exposed to, that's another technical complication. We did work, I have a collaborator here, Dimitri Abramson, who's working with some researchers at Tel Aviv University to actually mimic these microplastics, and we're doing some experiments and using in vivo testing to look at some of the potential human health effects. But, those are all issues that we're working through in terms of the research steps.
That's really interesting, and I'm looking forward to hearing about that. And then I guess we talked a little about weathering in the ocean, so we'll jump back to that. So for Kara, are there any, let's say, has there been an increase in microplastics floating on the sea surface over time? And then two, if we focus in on certain like oceanic gyres, are there any differences as far as distributions of microplastics?
Yeah, I think I'll start with the second question first. So my background is actually in physical oceanography. So that was kind of my entree into the field, was looking at where the plastics are accumulating. And I think it's been fully established that the microplastics that are floating at the sea surface are collecting in the center of oceanic gyres. So there's the boundary currents that we might be familiar with, like the Gulf Stream off of the East Coast or the Kuroshio Current. Those are really fast currents, but when you sort of go towards the center of the oceanic gyres, which span entire ocean basins in the 5 major North and South ocean basins, the currents sort of converge, they come together and slow down, and that's what creates these high accumulation zones, which is what the garbage patches are, right? They're big areas of high concentrations of floating microplastics. What was the second part? Oh, have the concentrations increased in time? So yes, probably. When we look at the data, I showed a graph of trends of beach debris over a decade on the US Northeast coast that was kind of a wiggly line. When we look at data in the North Atlantic, in that garbage patch accumulation zone in the center of the gyre, we do see evidence of an increase in concentrations, but it's actually fairly difficult to detect because there's a huge amount of variability within the ocean itself. So As an example, we had a research cruise that was trying to figure out how far east the garbage patch stretches towards Europe. All of our data had been in the Western North Atlantic. And on that cruise, we were doing sampling every 4 hours and towing a net and looking at what was in the net. And the net tow that had the highest concentration of plastics that we'd ever collected, it was 26 million pieces per square kilometer, which is huge. 4 hours later, there was a net that had very few plastics in it. So there's physical reasons why this is happening. The plastics can get mixed below the sea surface. It can have to do with what the sampling conditions are. So it takes some work to uncover these trends, and they're not always in the direction that you expect. But I think both from the North Atlantic and the North Pacific, we do see evidence of increasing concentrations with time, which I think makes sense just looking at the global production curve. The more plastics we make, the more are likely to leak into, percentage-wise, it might be the same amount lost to the environment, but there's more material out there that has that potential. So, yeah, hopefully that answered those questions.
Yeah, no, that's great. And I think it's also kind of getting into the next question about, how to collect or characterize. So I'd say that this is probably for everyone, but maybe we'll start with Chelsea too. So I think as the publications have increased, so have the questions about quality control and contamination control for other plastics that are used for research. I think it's a lot easier to get plastic things, but what are the ways that you all implement or have seen others implement reduction in plastic use during sample collection or characterization of microplastics, or even for doing health effect studies for whether that's in vivo or in vitro systems to reduce plastic exposure to reduce those confounding factors?
Imari, can I ask a clarifying question before I answer?
Yes.
You're asking what are the factors when we go out and we sample microplastic that may influence the answer that we get?
That is correct.
Okay. Yeah, so we've looked at this quite a bit in terms of sampling methods. We've also looked at it in terms of laboratory methods, but I'll talk about sampling methods for now because I think it's at top of my mind. So with the California work group, we have several smaller working groups to come up with what could be standardized methods for sampling in ambient water, in sediment, and in biota, as well as drinking water, but I won't focus on that. And then rivers. And I would say that for ambient water, we looked into this quite a bit because over the years, people have been using manta trawls. People are starting now to pump certain amounts of water in certain volumes. And we were really curious, what is the most important thing? Like, is it okay to use a manta trawl versus a pump? Do you get different answers? And if you look across the literature and you try to do a systematic review, it looks like you get different answers. And also the lower the volume, the higher the plastic. So we actually did a study at our experimental lakes area where we have a more controlled system where we put a manta with 53 micron mesh side by side with a pump with a 53 micron sieve. So we were targeting the same size. And what we found is that actually the answers were the same as long as you took enough volume to get a correct answer, meaning your limit of detection, you had to sample enough volume in order to be above the noise. So we know contamination is a big problem with microplastics. We have microplastics in the air. It's usually on our sample clothing. It can come from our nets. As long as you sample enough. In our case, what we found is that manta versus pump doesn't really matter. Now, I will say we weren't looking for microfibers. We were only looking for fragments. But I do think it's really important, and I think people are starting to realize this, that we absolutely have to sample enough volume. I think we've all been guilty, if we've done anything besides the manta, of not sampling enough volume, myself included. And I think we really have to figure out what is this limit of detection in your system, and then you can design your monitoring protocol. So that I think is something that really matters. There's lots of things that matter, but I think that would be a whole nother presentation. So I'll let others jump in.
Yeah, Kara, did you have any other additions to that? I know it's pretty similar water sampling.
No, I didn't know that Chelsea's group had done that comparison, and I think that's great and kind of makes my point from earlier that how we measure matters, and yeah, having a sample that has enough volume to be a representative sample, and that sort of nicely follows my description of, we had one tow that was 26 million pieces, another one that was a lot smaller 4 hours later. That you can't extrapolate either one of those and get the right answer. You need to be sampling enough to overcome the variability that's sort of the natural variability. I think also it's important to talk about laboratory contamination and the importance of controls and blanks, but I don't do that, so I'm going to lob it back over to Chelsea because I think that's also worth mentioning.
Sure, I can take it back or hand it over to Tracey, but I would say that yes, so in addition to the sampling methods, the laboratory methods are really important. You can use microscopy or you can do automated spectroscopy. I think if you're doing either, you have to be wary of the limits of detection in terms of size. But I think what Kara is getting at is that there's also a lot of procedural contamination during the extraction process and the quantifying process that also has to be taken into account. And I think when we started in this field, we weren't necessarily mimicking the analytical chemists who for years have had matrix spikes to look at recovery, have had lots of blanks both in the field and in the lab in order to compare their samples to. And I think we've learned that we need that too. And again, I've also been guilty of not always having these things, but today I'm really happy to see that a lot of these standard methods are incorporating these, and a lot of labs, including several of us in this room, have been trying to help create those recovery standards and help create those methods to inform better QA/QC.
That's great. Thank you both. And then Tracey, I know there's only a few quick minutes, but do you have like a 1-minute parting gift for QA/QC for human health studies?
I agree with Chelsea. The laboratory blanks is really the key because a lot of collection materials use plastics. That's true. So you have to have the collect a blank in the same materials and then use the method to kind of account for that in whatever you're measuring. But either way, I also agree larger studies are going to be better because just generally larger studies can help get the signal away from the noise. So that's why we need more research dollars in this important area. That's like my final recommendation.
Wonderful. Well, thank you all so much. I think we learned a lot today. And I guess just kind of a quick recap of what we've covered. I think we talked first about how plastics have been produced since the early 1940s. And the earliest instances of plastic pollution started typically in the marine environment around the 1960s and exponentially increased with more of those peer-reviewed publications noting larger quantities of plastic pollution in smaller and smaller size scales across the entire ecosystem. Now, the timeline for degradation and transformation has not been well defined, but our understanding of how some macro and micro scale plastics have a variety of impacts to biological organisms, including physical effects like dietary dilution or entanglement, to chemical effects like endocrine disruption. However, as we discussed, these impacts are not just isolated to environmental species. Human exposure to plastics can come from inhalation, ingestion, and even dermal exposure to plastics or plastic-associated chemicals. The toxicity evaluation is very different when we think about plastic-associated chemicals versus a pharmaceutical chemical before it's put into products and into the market. However, some of the post hoc observations or in vitro and vivo studies have shown so far that there are some suspected health impacts from plastics or plastic-associated chemicals, such as associated increases for some cancers or reproductive impairments due to these chemicals or even microplastics. So I want to thank all of our panelists for their thoughtful perspectives, and we extend our gratitude to the audience and the broader community for their engagement today. On May 13th through 14th, the Plastics Roundtable will be hosting a workshop on circularity in plastics. To register for this event, watch past webinar recordings and stay up to date on the activities of the Roundtable. Please navigate to our Roundtable website. And with that, we will adjourn. Thank you all again.